Welcome to our lecture on the resting membrane potential. Today, we're going to explore a fascinating secret hidden inside your body. Every single nerve and muscle cell you have acts just like a tiny microscopic battery. We will uncover how this battery works, why it is absolutely essential for your survival, and the elegant mathematics that govern its behavior. Let us dive in. To begin our journey, let us think about a simple AA battery you might put into a television remote. Just like that battery, your body's cells have a positive end and a negative end. When a battery is just sitting on a table, it holds a stored electrical charge, waiting to be used. In the human body, this waiting state is called the resting membrane potential. Think of it like a stretched rubber band. It is not moving right now, but it possesses a massive amount of stored energy ready to snap into action the moment it is released. Now that we have our battery analogy, let us look at the formal scientific definition. The resting membrane potential is the electrical voltage difference that exists across the plasma membrane of an excitable cell under resting conditions. For a typical human neuron, this baseline value is approximately -70 millivolts. That negative sign is incredibly important. It tells us that the inside of the cell is negatively charged relative to the outside. This specific voltage is the precise baseline that the cell works very hard to maintain when it is at rest. Understanding what this voltage is leads us to ask how it is created. This electrical difference is generated by an unequal distribution of charged particles, called ions, across the cell membrane. The main players in this process are sodium ions and potassium ions, along with large negatively charged proteins that are permanently trapped inside the cell. Because the cell membrane is semi-permeable, it allows some of these ions to leak back and forth. This delicate leaking perfectly balances the electrical and chemical forces to maintain that -70 millivolt baseline. Before we look at the math, we must understand why the body spends so much energy maintaining this state. This resting potential is the absolute foundation for all communication in your nervous system and all movement in your muscular system. Without this stored electrical energy, your brain could not send a signal to your hand to move away from a hot stove, and your heart muscles could not beat. In medicine, understanding this is crucial. For example, local anesthesias used by dentists work specifically by altering this membrane potential to stop pain signals from ever reaching your brain. Now that we know why this matters, let us explore how this voltage is calculated mathematically. If the cell membrane were permeable to only one specific ion, that ion would move until its chemical gradient is perfectly balanced by its electrical gradient. The exact voltage where this balance occurs is called the equilibrium potential. We calculate this using the Nernst equation. E ion represents this equilibrium potential, R is the gas constant, T is temperature, Z is the electrical charge of the ion, and F is the Faraday constant. The terms in the brackets represent the concentration of the ion outside versus inside the cell. If the concentration difference increases, the voltage required to balance it also increases. This complex formula we just saw leads us to a much more practical version. Because human body temperature is relatively constant at 37 degrees Celsius, we can combine all those constants and convert the natural logarithm to a base 10 logarithm. For a monovalent ion, the formula simplifies beautifully. The constant 61 represents the combined thermal and physical constants and gives our result directly in millivolts. The variables still represent the outside and inside concentrations. This simplified version is what doctors and physiologists use most often. It clearly shows that if the concentration is higher outside the cell, the ratio is greater than 1, and the potential will be positive. Now that we have our simplified formula, let us see this with a concrete example for potassium. We are given an inside concentration of 150 millimoles per liter and an outside concentration of 5. We plug the outside concentration of 5 into the numerator and the inside concentration of 150 into the denominator. Dividing these gives 0.0333. Because this is less than 1, its logarithm is a negative number, specifically negative 1.477. Multiplying this by our temperature constant of 61 gives us our final answer. The equilibrium potential for potassium is approximately negative 90 millivolts. This means potassium leaving the cell leaves behind a strong negative charge. Having calculated potassium, let us apply the exact same approach to sodium. For sodium, the concentration is much higher outside the cell than inside. We are given an outside concentration of 150 and an inside concentration of 15. We place 150 in the numerator and 15 in the denominator. Dividing these gives us exactly 10. The base 10 logarithm of 10 is simply 1. We multiply this by our constant of 61. Our final answer is positive 61 millivolts. This makes perfect intuitive sense. If positively charged sodium rushes into the cell down its gradient, it will make the inside of the cell highly positive. So, we have successfully calculated the equilibrium potentials for both ions. This brings us to a crucial physiological proof. Let us set up our known values. First, we know from direct measurement that the actual resting membrane potential of a typical neuron is negative 70 millivolts. Second, our math just showed us that potassium wants the cell to be at negative 90 millivolts. Third, sodium wants the cell to be at positive 61 millivolts. We have a tug of war happening across the cell membrane between these two ions. In the next step, we will see who is winning this electrical tug of war and what that proves about the membrane. Continuing our derivation, let us look at the final result. The actual resting voltage of negative 70 millivolts is significantly closer to potassium's negative 90 millivolts than it is to sodium's positive 61 millivolts. In physics, the overall membrane potential is always pulled closest to the equilibrium potential of the ion that can cross the membrane most easily. Because the actual voltage sits so close to potassium's ideal voltage, we have mathematically proven a fundamental principle of neurophysiology. The resting cell membrane is far more permeable to potassium than it is to sodium. Potassium is the dominant force in the resting cell. Now that we have proven the math, let us visualize these mechanics in action. On screen is a detailed schematic of the cell membrane. At the top is the extracellular fluid, packed with sodium ions. At the bottom is the intracellular fluid, rich in potassium and large negative protein anions. Notice the channels embedded in the membrane. The potassium leak channels allow potassium to easily exit the cell, which we proved is the dominant movement creating the negative charge. You can also see the sodium-potassium pump, actively using energy to push 3 sodium ions out for every 2 potassium ions it brings in. Together, these structures maintain that perfect negative 70 millivolt reading shown on the right. To conclude our lecture, let us review the key takeaways. First, the resting membrane potential is the vital stored electrical energy of a cell, typically sitting at -70 millivolts. Second, this battery-like state is maintained by the unequal distribution of ions and the specific permeability of the cell membrane. Third, we can use the Nernst equation to calculate the exact electrical force generated by any ion's concentration gradient. Finally, because the membrane is highly permeable to potassium, potassium's outward movement is the primary driver of the cell's negative resting state. You now understand the fundamental electrical foundation of human physiology.